The next presentation is by John Mascarello, who is a PhD at CMC Biologics. His presentation, Continuous Disposable Multi-Column Chromatography, Emerging Technology to Enable More Efficient Clinical Downstream Processing. So without further ado, I'd like to introduce John.
All right, well, I'd like to thank everybody for coming to hear my talk. I'm going to talk about something that I think is a pretty exciting advancement in the field and something that I'm excited for CMC Biologics to be offering for our clients. It's really talking about disposable downstream processing and what we can actually do to implement some disposable technologies in downstream processing. So I realize that nobody's probably going to be able to read these quotes, but I think anyone that's been in the field kind of realized that there's been a lot of innovation. And a lot of that innovation, we've actually seen that in the upstream part of process development. We've seen single-use bioreactors, we've seen harvest technologies that are disposable. We've seen a lot of really good advancements. And a lot of those have led to increased titers and a lot of the benefits we're seeing today. But you'll see a lot of these quotes and a lot of the people are talking about what's going on with downstream. Why haven't we seen anything really come forward that talks about innovation in downstream and how we can move away from just regular batch chromatography. So here's just a schematic of a traditional batch process, nothing too crazy. Bioreactor, 3 chromatography columns, some nanofiltration, some viral inactivation, filling it into the drug substance. It's the middle part of this process that's problematic, right? It's the chromatography. Like I said, we have a harvest, we've implemented some disposables, upstream harvest. We very commonly use single-use mixers for product holds and buffer holds. We'll use nanofiltrations. The real difficulty in finding something that's disposable is around chromatography. And the tough part of this is chromatography is such a powerful tool for purification. We can't really get rid of it. But the problem is you need to buy a lot of it in order for you to purify the batches that you have. And that's difficult to figure out a way to go ahead and get that to be disposable. And that's what I'm going to talk about in my talk. So what I'm going to propose and what CMC is offering to implement disposable downstream processing is a technology called multi-column chromatography. And I'll talk about this in a little more detail in the next couple of slides. So first we'll talk about what is batch chromatography. Batch chromatography, you go step by step by step. You have to equilibrate your column, you have to load your column, then wash it, elute it, and clean it. All those things take a lot of time, and you're doing them in series. And there's a lot of that stuff that's really not value added. Equilibrating the column really isn't that much value added. And then traditionally, when you're doing it in a batch, you tend to somewhat underload the column because you want to avoid any loss of your product that might come out when you're loading it. So you typically are very conservative in how you load your column. Multi-column chromatography, as the name implies, you have multiple columns. And I'm going to go through a schematic down here, and hopefully you can see it in the slide deck. So in this situation, we have 3 columns, but you can actually go up to any number of columns. 8 is kind of the max end. And you're always loading, washing, and eluting columns kind of in parallel. So in this situation, we have 3 columns. 2 of them are connected and will be loaded at the same time. So this would be column A and column B. As I mentioned, a lot of times when you do chromatography, you want to be very conservative not to lose any product. In this situation, you have a column after the primary column that will capture any product that might come through. So you're not having a risk of losing product because it'll be captured by that second column. And this initial situation, the 3rd column isn't doing anything. Once you get to the point where the first column is loaded appropriately, you go ahead, you take that out of the load stage, you wash it, elute it, clean it, equilibrate it, and at the same time, you have your other 2 columns which are now connected and are being loaded at the same time. And this will just go around and around in a carousel where you take each column out once it's reached the load that you want, start doing the wash, elution, and you'll start loading other columns. What you can see here is a couple benefits. First of all, you're always loading a column. So a lot of that dead time of just equilibrating all those things, you're always loading columns. One thing that's really interesting, thinking about continuous processing, is this is directly applicable to perfusion. Perfusion in your bioreactor, you're constantly taking product out of your tank, and so you can now interface this technology and continuously load a column in this multi-column scenario. The other thing that's very nice about this, as I mentioned, is you don't have to worry as much about losing your product. You can have that second column afterwards that captures any product that would be coming out of that first column because you're trying to do it very rapidly. So this is kind of the fundamentals, and I'm going to talk about some of the advantages this technology has relative to kind of the standard traditional batch processing. So I'll focus in the two aspects. Clinical, which is going to be the focus of what I'm going to talk about today, but there's also commercial applications. So for clinical, what we hear a lot from our clients is the problem statement, hey, look, I have to buy a whole bunch of resin that I'm not going to use that much, right? I might need 1 or 2 batches in order to make my material to go into the clinic and see if I'm going to have proof of concept. And there's a high rate of attrition, so oftentimes I spend a lot of money on resins that I'm not using that much. The advantage of the multi-column chromatography is you can actually rapidly go through those cycles that I showed in the previous slide, and you can actually do, in the time that you have in your manufacturing suite, on the order of about 50 cycles on each column. So you're getting to the point where you're actually using close to the lifetime of your column. The other aspect is you can actually, by doing these things very rapidly, as I mentioned, you're always doing something productive on your column, whether loading a column or eluting a column. It has the potential to use 90% less resin than traditional batch. And I'll go through some of a case study around that, and I'll talk a little bit more detail around that. But that's a significant benefit for people that just want to make material to go into the clinic to see if this is going to be effective or not, and not want to spend hundreds and hundreds of thousands of dollars in resins. These things in combination, the fact that you're cycling close to the lifetime of the column and you're using very small amounts of resin, this really enables this to be disposable. You can now get to the point where you're buying pre-packed columns. You don't have to worry about doing all this column packing in your manufacturing suite. You can buy pre-packed columns, have them be part of your supply chain, and have that be a very seamless integration into how you handle your columns, and not have to worry about storing them and packing them. As I mentioned, there's also applications for commercial. As I mentioned, you have these 2 columns that are connected in series. You can now make sure that first column, you're loading all of the active sites that you have there. In traditional batch processing, you're very conservative because you don't want to have product come out at the end. You lose a gram of product at a commercial stage, that's tens of thousands of dollars that you could have probably sold that product for. This allows you to actually capture any product that would be lost on the subsequent column to make sure you use all of the binding sites of that first column. That actually leads to having you bind more mass per liter resin, which you'll see as a decrease in your commercial cost of goods. So I'll have a little bit more of a discussion about that in the next slides. So these are just some of the diagrams that you can make with this multi-column chromatography. On the y-axis is the relative amount of resin that you use relative to the batch process. So you can see the batch process is up at 1. The x-axis is the amount of grams that you produce per liter of resin. So 100% would be the batch process, and that's why the reference point's at 1 and 100%. Like I said, in commercial, you would tend to want to operate in a way that you're pushing as many grams onto the resin as possible. And you can see this technology on this line here. You would use 60% of the resin of a traditional batch, but all of that resin you would be actually making 40% more grams out of that per liter of that resin. What I'm really interested in is this blue box, the disposable range, which again, you can use about 10% of the resin that you would use for a traditional batch process. And I think that's very attractive to people trying to get into the clinic. The one thing I want to emphasize is this isn't something that takes a ton of time. What you do in order to do this is you actually generate some standard breakthrough curves for your chromatography, as you can see in the bottom of the slide, that will allow you to figure out things about your static binding capacity, but also allow you to think of some of your mass transfer coefficients so you can understand the mass transfer of how protein goes onto the resin. Once you generate those breakthrough curves, everything else is computer modeling. You're actually going to implement the dynamic binding capacity and the mass transfer coefficients, use that to interface with the system that we use from Pall that will actually develop the column geometry, the number of columns, the cycling time to meet the needs that you want to optimize to. And oftentimes that's minimizing resin. You can play around with that tool, get the configuration that you want, and then you can just test it out in your program, and we've shown very good comparability in terms of the in silico predictions and what we actually see in production. The other thing that's important, I just want to show you, this is actually probably something that you can't see in too great detail, is it's working very robustly. There's showing excellent process consistency. One of the concerns I would have as a process developer is I'm showing differences between column 1, column 2, and column 3. I'm showing a difference over time that my first cycle looks different than my 30th cycle. In this situation, this is actually doing some multivariate analysis around the elution profiles, both by UV and pH, and doing some principal component analysis to see if anything is changing over time. In this situation, we ran 3 columns, each column for 30 cycles, and the principal component analysis shows there's no systematic differences, there's nothing that's changing with cycle number, and there was no differences between column to column. So that means it's a very robust technology that we know we're not going to see things change along the way. So now I'm going to go forward and show you a case study that we want to present really around how this technology can be used in a clinical stage. So I'm just going to have some assumptions. Everybody's assumptions could change. And this is just one model, and there's many, many models that you could develop. So in this situation, we have a 2,000-liter bioreactor, 3 gram per liter titer process, 80% downstream yield, and due to the manufacturing site, we want to complete our downstream in 5 days. Pretty standard stuff. Something that we offer, and I think a lot of people offer, is for molecules that are very well understood, we can go right into GMP, maybe not the need to do an engineering run, and with this type of process, with the yields and the titers that we have, we're going to make on the order of about 5 kilograms. For a lot of projects, that's enough to get through tox, phase 1 and phase 2, so you might only need that one run really to get into the clinic and show proof of concept. So traditional batch processing is resin-based, protein A, CEX, AEX for a monoclonal antibody. These are kind of standard prices that are out there depending on which resins you like and who you like to buy from. Disposable, again, we would be using single-use pre-packed columns for the protein A and CEX and run them in multi-column chromatography mode. And then we're going to look into a single-use membrane for the AEX step. And we'll do some analysis and show how this actually works. So the big bang for your buck, obviously, is on protein A. In this assumption that I have in this model, I was very conservative and chose an 8-hour operating time, so just 1 day in your manufacturing suite. As you know, with a 3-column process, there's often potential to do 2 days worth of protein A in your manufacturing suite. And that's just going to make the numbers show up to be better. As I mentioned, you're not really in this scenario, you're not loading a lot more onto your column, but you're going a lot faster. You can see we're actually going at a 1-minute residence time in the MCC mode versus a 3-minute residence time on your batch process. And you can actually see the difference that I was talking about. In this situation, we've gone down from 60 liters of protein A resin down to 11. And we're going to do that in 5 columns that are 20 centimeters in diameter and 5 centimeters in height. You can see that we use 6 columns in the multi-column chromatography manner, and then we're going to cycle them in this situation only 15 times. There's things that we can do to make that go quicker, but just through the process that we have, it's 15 times. There's a couple more things down there, but I think the main take home is at the bottom. In this scenario, with even 4 cycles on your batch chromatography, you would have to spend close to $1 million in protein A resin. With the multi-column chromatography, you're spending on the order of about $200,000. So about a 75% decrease in the cost when you implement this technology. As I mentioned, we're working with Pall. That's the system that we like. That has the potential to go up to 8 columns, which adds a lot of flexibility. There's potential to get even further savings by increasing the amount of time that you can do this operation in manufacturing. And then, of course, process development can do things to even optimize this a little further. The other example I want to talk about is using membrane, AEX membranes instead of resins. This has been around forever. This is something I think most people have experience with. But this is just, again, in the vision of a purely disposable downstream process, another way that you can implement a disposable technology. I think it's pretty well established that the benefits of membrane chromatography are really that you're able to load significantly higher on your membranes relative to your resins, and that provides you that cost of goods benefits. In this situation, you can see we will load about 150 grams per liter on a resin, but we can load up to about 2,000 liters on a membrane and still get comparable yield and product quality. And that results in about a $90,000 savings from a cost of goods standpoint, and again, something that's disposable. There's things that we haven't talked about, but these technologies also have benefits in terms of buffer consumption and decreasing the amount of buffer that you would need to use. So all told, in this scenario, going into the clinic, you're going to save on the order of about $1 million. Right? The traditional, if you have to spend your $900,000 on protein A, $250 on CEX, and about $118 on AEX, you can see those numbers are reduced pretty significantly in the disposable case. It's worth mentioning, obviously, in the traditional case, if you ever needed to do resupply runs, you have all those membranes or you have all those resins. You don't need to repurchase that. But again, as titers have gone up and the advancements that we have, the amount of material that we're getting per one lot is often enough to get into the clinic and really show if that molecule has potential and is worth more rounds of process development. Again, increasing the amount of time you can do in manufacturing is just going to make these benefits that much better. As I mentioned, there's additional benefits that I haven't put in dollars to associate with in terms of buffer consumption, the decrease in prep time and clean time because they're disposable rather than something that needs to be cleaned, the ease of the supply chain and purchasing pre-packed columns, and again, the lack of resins to store. So one thing to also note is the benefits get more beneficial as the titers go up. As the titers go up, you get more and more mass per lot, so that just decreases the chances you'll need to do a resupply campaign. And as your titers go up, it actually allows you to cycle those columns in the multi-column chromatography system faster and faster to increase the number of cycles that you do per each column. So just overall conclusions, this is a technology that we're really excited about and we're really excited to offer our clients. It shows, hopefully, that it's a cost-effective way to implement disposable chromatography and downstream processing. Again, I think the problem is really with traditional chromatography, the limited number of lots that you do at a clinical stage, you only use about 5 to 10% of your lifetime during clinical manufacturing. And this multi-column chromatography setup has the potential where you can rapidly cycle your column without impacting performance, so you can use significantly less resin and use that resin to a high number of cycles. It has the potential to decrease your clinical cost of goods for your protein A resin and your batch resins by about 90%, which is obviously a huge, huge saving that we can offer our clients. And again, we can use pre-packed columns, so we're really making very efficient for our manufacturing folks with not having to pack a whole bunch of columns over and over again. And then hopefully I showed you that case study where we used disposable technologies, both in terms of protein A, CEX, and AEX, and showed that we can save on the order of about $1 million at the clinical stage for our clients. So some of the acknowledgements, Michelle and Chase were in the lab putting together the data that we are actually able to present in the case studies. And then again, we're working with Paul, who's been a great collaborator in getting this technology up and running here at CMC. And with that, I'll take any questions.